In this mini tutorial we're going to think about the cells which are present in the innate immune system, and there are three broad classes of innate immune cells. We have the so-called antigen presenting cells, or APCs, and it's the role of antigen presenting cells to take up antigen found in pathogens, to process it in a certain way, and then to present it to cells of the adaptive immune system so that they can then mount a response. And APCs are also important for the activation of other cellular elements in the immune system. We have the phagocytes, and as you are aware, phagocytes are important for taking up material and destroying it, and in some cases certain phagocytes can act as antigen presenting cells themselves. And the third class are the natural killer cells, the NK cells, and these cells have a role in protection against viral infections and cancer. So let's consider antigen presenting cells, and the type of antigen presenting cell that we're going to think about are the APCs which live in the skin. So if you go back to one of the previous tutorials where we did a quick picture of the skin, so there's the basement membrane of the skin epithelium. Here are our skin cells, and I'm purposely spacing them out a bit more than they would normally be. So this is the epidermis of the skin, the surface layer of the skin, and these red blobs are the skin cells. And within the epidermis of the skin there are antigen presenting cells, and I'm going to draw one of these antigen presenting cells in blue. And what the antigen presenting cells in the skin have are these very long extensions. In fact they're called, another name for these is dendritic cells, from dendros meaning kind of branch, like a tree branch. And you can see how these branches insinuate themselves all through the skin cells. And what the antigen presenting cells have on their surface are various receptors which are constantly sampling the fluid in between the cells of the skin, looking for pathogens. Now, if the antigen presenting cell detects a pathogen, which we'll use a green blob to represent here, if it detects a pathogen, then what it does is the APC ingests that pathogen, it phagocytoses it into its cytoplasm, and then the antigen presenting cell itself migrates out of the skin to a lymph node. And within the lymph node, the antigen presenting cell is able to activate other cells of the immune system, for example B cells or T cells. So it's able to tell the rest of the immune system that there is a pathogen present, in this case in the skin, and it's able to activate relevant immune cells to mount a response. So APCs are very important in initiating an immune response. A second class of cell present in the innate immune system is the phagocyte, and a classic type of phagocyte that we typically think about is the neutrophil. So here's a neutrophil, and you can always tell a neutrophil quite easily by the fact that its nucleus is lobed. There are multiple lobes in the nucleus of a neutrophil. So it has this many-lobed nucleus. And what neutrophils are involved with, as you know, is endocytosis or phagocytosis of typically bacterial pathogens. So as we said, this is a neutrophil, and they are involved in phagocytosing primarily bacteria. And it is frequently neutrophils which are the first cell type on the scene during the process of an infection. And what neutrophils have on their surface, once again, is a whole array of different types of receptor for all sorts of pathogens. And these receptors recognise, for example, polysaccharides on bacterial cell walls, and they will, in a non-selective way, take up pathogens. So, for example, here might be a bacterium which our neutrophil has recognised through the green receptor on its surface. It takes that bacterium up and then into an endocytic vesicle. Then a lysosome will fuse with that vesicle and destroy the bacterium. Now, a second type of phagocyte in the innate immune system is the macrophage. Now, macrophage is actually developed from cells which are found in the circulation, and these cells are found in the blood. Quite big cells, once again with fairly large nuclei like this, and these cells which are found in the blood and are the precursors for macrophages are called monocytes. Now, monocytes are able to leave the circulation, so they're able to cross blood vessel walls and reside within various body tissues. And when a monocyte leaves the circulation and then sits within whatever body tissue you might want to consider, whether that be skin or gut or brain or whatever, that monocyte, once it's left the circulation, becomes known as a macrophage. So macrophages are simply monocytes which have left the circulation and started to reside within a specific tissue. Now, once again, like the neutrophil, the macrophage is a phagocyte. It has a number of receptors on its surface, allowing it to recognise foreign material, to endocytose it and then destroy it using lysosomes. But in addition, what macrophages can also do is they can actually act as antigen presenting cells. So the pathogen that they've taken in and chewed up, they can then go and show that to other cells of the immune system within a lymph node, just like the APC that we considered previously. The final class of cell which I want you to think about in the innate immune system is the natural killer cell, the NK cell. And NK cells are quite interesting, really, and they have very important roles in immunity against viral infection, and also they have a role in destroying cancer cells. And the way they do this is quite interesting, really. If we consider a normal cell, so this is any cell you might care to think of in the body, a normal cell. That normal cell normally, as you know, expresses a whole load of different cell surface molecules, proteins, which might be receptors, or other factors on the surface of the cell. So normal cells express a whole load of different proteins on their surface. And if the NK cell comes along, so we can draw an NK cell here, it has receptors upon its surface which recognise these so-called normal cell surface receptors on other cells. So the NK cell has receptors which recognise these normal receptors. And if the NK cell's receptors are activated by the presence of normal proteins on the surface of our cell, the NK cell does nothing. So it's not interested in cells which express their usual full complement of surface proteins. However, if we have a cell which is abnormal for whatever reason, whether that be a cell which is virally infected or a cancer cell, what we know is that these infected or cancerous cells frequently display different levels of surface proteins. And in fact, sometimes they have much lower levels of surface proteins than would normally be seen. And in this instance, our NK cell comes along, and its surface receptors here are not engaged with any of the normal proteins found on the surface of our abnormal cell. And it's the absence of these normal proteins which activates the NK cell. And when the NK cell is activated, it does many things, but basically all of these things have the end result of killing the diseased cell. So for example, one thing that the NK cell does is it releases molecules called perforins, and as I'm sure you could work out from the name, perforins are involved in perforating the cell membrane of our diseased cell. So they put holes in the cell membrane like this. They permeabilize the diseased cell. And that does a number of things, one of which obviously it messes up the membrane potential of the diseased cell. Substances leak out and leak in, and so the cell is going to be under considerable stress from this attack on its membrane. Secondly, what the NK cells do is they release molecules which actively stimulate apoptosis, or programmed cell death, within our target cell. And one such molecule released by NK cells is called granzyme. And what granzyme is able to do is cause our target cell to kill itself by the process of apoptosis. So in summary, if an NK cell discovers that the levels of cell surface proteins in a target cell are altered, it can release perforins to permeabilize the cell membrane, and granzyme and other molecules to cause that cell to actively kill itself. So that concludes our overview of the cells of the innate immune response.